Welcome to our channel, Mootechie. We're glad to have you here as we dive into the fascinating microscopic world. Learn microbiology with clean visuals. Presentation Overview. Principles and general properties of microbial classification. This is the framework we use to organize the immense diversity of microbes in a logical way. Exploring the polyphasic approach to identifying and categorizing microorganisms through evidence-based methods. This means we don't rely on just one piece of evidence. We combine multiple lines of data for the most accurate picture. Principles and logic. Understanding taxonomy, systematics, and nomenclature rules. These are the foundational rules and goals that guide the entire classification process. Genotypic analysis, DNA-based methods and evolutionary relationships. This involves looking directly at the genetic code to see how closely related different microbes are. Phenotypic traits, morphology, physiology, and biochemical capabilities. These are the observable and measurable characteristics of the organism itself. Chemotaxonomy, chemical markers in cell walls and membranes. This analyzes the unique chemical building blocks that make up a microbe's structure. The diagram shows these three major evidence types: genotypic, phenotypic, and chemotaxonomic, all feeding into the central goal of accurate classification. Let's build a strong foundation by understanding the basics. Foundation. Introduction to microbial classification. Purpose of classification. Enables logical organization, clear communication between scientists, and facilitates new discoveries. By having a standardized system, researchers worldwide can accurately discuss the same organism and build upon each other's work. Key terminology. Distinguishes taxonomy from grouping, systematics from evolutionary history, and nomenclature from naming. It's helpful to remember taxonomy is about creating the groups, systematics studies their evolutionary relationships, and nomenclature gives them official names. Taxonomic hierarchy organizes life into ranks from broad domain down to specific species and strains. Think of this like an address, going from a large country down to a specific street number. Standardized naming. Follows strict codes like ICNP for prokaryotes and ICN for fungi and algae to ensure universality. These international codes prevent confusion, ensuring that Escherichia coli refers to the exact same bacterium everywhere in the world. The pyramid graphic illustrates this hierarchy visually, starting broadly with domain, then narrowing down through phylum, class or order, family or genus, and finally to the most specific level, species. The taxonomic rank narrows down from general groups to specific individual types. This funnel-like process helps us precisely pinpoint any organism in the vast tree of life. Now, let's look at the guiding ideas behind this system. Core principles. Principles of microbial classification. Evolutionary relationships. Reflects natural classification by prioritizing phylogenetic history over simple resemblance. We group organisms based on their shared ancestry, not just because they look alike, which gives us a much truer picture of their relatedness. Polyphasic framework. Integrates phenotypic, genotypic, and chemotaxonomic data for robust consensus. Relying on only one type of data can be misleading, so using all three together creates a stronger, more reliable classification. Type strains. Anchors valid names to permanently preserved type strains acting as living reference standards. When a new species is named, a specific sample is designated as the official reference example that all others are compared against. Reproducibility. Ensures taxonomic descriptions are reproducible and standardized across international laboratories. Any trained scientist following the methods should be able to identify and classify the same organism the same way, which is crucial for scientific consistency. The diagram on the right shows 3 circles representing phenotype, genotype, and chemistry, all overlapping in the center to form a valid species. The polyphasic approach combines multiple lines of evidence for accurate classification. This Venn diagram is a perfect visual for the polyphasic concept. True, valid species definition sits at the intersection of all these key data types. With the principles in mind, what specific traits do we actually look at? Here's an overview. Properties overview. General properties used in classification. Microbiologists utilize a combination of 5 distinct property categories to accurately identify and classify an organism. These categories give us different lenses through which to examine and understand a microorganism. Morphology. Physical form, including cell shape, arrangements, spores, flagella, and colony appearance. This is often the first clue, like recognizing a friend by their height, hairstyle, or the way they walk. Physiology. Growth requirements such as temperature, pH, salinity, and oxygen tolerance. Understanding where and how a microbe thrives tells us a lot about its identity and ecological role. Biochemistry. Metabolic capabilities, specific enzymes, carbon source utilization, and fermentation. This reveals the microbe's chemical toolkit, what it can eat, what waste it produces, and how it generates energy. Genetics. Analysis of DNA/RNA sequences, G+C content, and genomic comparison. This is the ultimate blueprint, allowing us to compare genetic codes directly to determine evolutionary kinship. Ecology. Environmental niche, host interactions, pathogenicity, and symbiotic relationships. Knowing if a microbe lives in hot springs, causes disease, or helps a plant grow adds critical context to its biological story. Let's zoom in on the first 2 classic categories, what we can see and how the organism lives. Morphological and physiological traits. Cellular morphology. Identifying cell shape, size, arrangements such as chains or clusters, and visible colony appearance on agar. Under the microscope, a rod-shaped bacterium arranged in a chain tells a different story than a cluster of round cells. Staining and structures. Differentiation via Gram reaction, positive or negative, presence of endospores, capsules, or inclusion bodies. The Gram stain is a fundamental first test, instantly dividing bacteria into 2 major groups based on their cell wall structure. Motility mechanisms. Observation of movement types, such as swimming via flagella, gliding, or swarming patterns on surfaces. Whether and how a microbe moves can be a key diagnostic feature, observed under a microscope or on special agar. Growth conditions. Tolerance ranges for environmental factors, optimal temperature, thermophiles, pH levels, and salinity, halophiles. Understanding where and how a microbe thrives tells us a lot about its identity and ecological role. Oxygen relationships. Classification based on oxygen needs, obligate aerobes, facultative anaerobes, or microaerophiles. This tells us if the organism requires oxygen to live, can live with or without it, or is even poisoned by it. 1. These observable traits form the traditional foundation for initial microbial identification in the laboratory. For over a century, these have been the first and most accessible tools for a microbiologist starting to identify an unknown culture. Moving deeper, we explore the metabolic fingerprints of microbes. Phenotypic characteristics. Metabolic and biochemical traits. Substrate utilization. Ability to use specific carbon or nitrogen sources and distinct energy strategies for growth and survival. A microbe that can only grow if you provide the sugar lactose is biochemically distinct from one that can use many different sugars. Enzyme assays. Detection of key enzymes like oxidase or catalase using standard biochemical panels like API or biological. Simple tests, like seeing if a bubble forms when you add hydrogen peroxide, reveal the presence of specific enzymes. Metabolic pathways. Differentiation based on fermentation versus respiration profiles, and distinct gas or acid production. Whether it produces acid, gas, or a particular smell from a sugar can pinpoint its identity in a standardized test strip. Antimicrobial patterns. Analysis of susceptibility or resistance to various antibiotics, providing supportive classification data. While primarily used for treatment, the unique pattern of which antibiotics stop a bacterium's growth can also aid in its identification.
Secondary metabolites. Production of unique pigments, toxins, or other distinctive chemical compounds used for identification. The vibrant color of a bacterial colony or its ability to produce a specific toxin can be a clear, visible calling card. Biochemical fingerprinting reveals the functional capabilities hidden within the microbial genome. These tests essentially ask the microbe a series of yes or no biochemical questions, generating a profile that is often unique to a species. Now, let's look at the modern revolution in classification, analyzing the genetic code itself. Genetic characteristics, DNA-based methods. Modern microbial classification relies heavily on DNA sequencing and genomic analysis to determine precise evolutionary relationships. This is like moving from comparing outward appearances to comparing the detailed instruction manuals written inside each cell. 16R RNA gene, the universal standard for identification and inferring phylogeny at the genus level. This gene evolves slowly and is present in all bacteria, making it perfect for placing an unknown microbe on the family tree. Species metrics. Precise genomic thresholds like ANI greater than 95% and DDDH greater than 70% define species. These are numerical, objective benchmarks. If two genomes are 95% identical, they are considered the same species. MLSA or MLST. Sequencing multiple housekeeping genes offers high resolution for strain-level typing. By looking at several stable genes, we can distinguish between very closely related strains, crucial for tracking disease outbreaks. Phylogenomics. Constructing trees from whole genome core genes provide robust evolutionary frameworks. Instead of one gene, we use hundreds of essential genes from the entire genome to build a much more reliable and detailed evolutionary tree. Genome features. Signatures like G+C content, gene synteny, and codon usage patterns. Even broad genomic characteristics like the percentage of G and C nucleotides in the DNA can be a useful first hint about an organism's identity. Our third major line of evidence comes from analyzing the chemical makeup of microbial cells. Chemical markers. Chemotaxonomic characteristics. Analyzing specific chemical constituents of the cell provide robust, stable markers that are essential for classification at the genus and species levels. While genes provide the instructions, these chemicals are the actual building blocks, offering a very stable and informative profile. Cell wall. Composition of peptidoglycan, variation in DAP isomers, and presence of teichoic acids. The precise structure and chemistry of the cell wall is a major divider, most famously between gram-positive and gram-negative bacteria. Membrane lipids. Analysis of fatty acid methyl esters and polar lipid profiles. The specific types of fats present in the cell membrane create a unique lipid fingerprint for many groups of bacteria. Respiratory quinones. Types of electron carriers like ubiquinones versus menaquinones. The molecules a microbe uses to generate energy in its respiratory chain are often specific to certain taxonomic groups. Isoprenoid ethers. Distinctive archaeal ether lipids, isoprenoid chains, sterols, and hopanoids. These unique branched chain lipids are a hallmark of archaea and some bacteria, clearly setting them apart. Polysaccharides. Specific cell envelope sugars, diagnostic polysaccharides, and exolipids. The unique sugars and complex carbohydrates on the cell surface can be highly distinctive, like a chemical ID badge. Let's bring all these concepts together and review the most important points. Summary and key takeaways. Effective classification requires integrating diverse biological data into a cohesive framework. No single method is perfect. The true power comes from weaving all the evidence together. Polyphasic approach. Modern classification is evidence-based, integrating multiple datasets rather than relying on a single method. This is the central, overarching theme of everything we have discussed. The Holy Trinity. Reliable taxonomy combines phenotype, appearance, genotype, DNA, and chemotaxonomy, chemistry. These three pillars support a stable and accurate understanding of what a species is. Type strains. Naming and identification must be anchored to valid reference cultures to ensure reproducibility and stability. They are the official representatives, the gold standards to which all other isolates are compared. Genomic revolution. Whole genome sequencing and ANI metrics are reshaping boundaries, becoming the new gold standard. DNA sequencing is providing unprecedented clarity and is increasingly the definitive tool for classification. Contextual relevance. Ecological niche and clinical presentation provide essential meaning to biological clusters. Knowing a microbe's genetic group is important, but knowing if it lives in soil or causes pneumonia gives that information its full significance. And that brings us to the end of our overview. We hope you enjoyed this overview of microbial classification. Questions? We are here to help. Subscribe to Mootechie channel for more. Thank you for your curiosity and engagement. We hope this foundation helps you see the incredible order within the microscopic world, and we look forward to exploring more with you next time.